Hi, everybody, and I want to welcome you to the Center for Brain Health, where we're taking a deep dive into the most exciting new developments in brain research. My name is Bart Ritma. I'm a professor in the at UT Dallas in the School of Behavioral and Brain Sciences, and I'm do research here at the Center for Brain Health. I want to mention that we will have a few minutes at the end to answer questions, and for the virtual audience, please use the Q&A function to register your questions. So for those of you who don't know much about us, the Center for Brain Health is a cognitive neuroscience research center at the University of Texas at Dallas, where we've dedicated the past 3 decades to exploring neuroplasticity and the brain's amazing potential to get stronger and work better. And now I'd like to introduce our speaker, Dr. Monroe Turner. It's a real honor to be presenting him because he did his doctoral work in my lab investigating how changes in blood flow properties of the brain are related to changes in cognitive performance, and made some real great strides in my lab, publishing around 24 papers, several of those that he was first author on. And he now works with Dr. Jeffrey Spence, who's the director of biostatistics here at the Center for Brain Health, and he's actively processing and analyzing data from the very ambitious neuroimaging arm of the Brain Health Project. So without further ado, let me give you Monroe Turner.
Thank you so much, Bart, for that magnificent introduction. Hi, everybody. When Dr. Ritman just said deep dive now, I think that today I'll be able to deliver on that. So we are going to go on a fairly deep dive, and we're going to be talking about enhancing brain health with functional MRI, insights and lessons. And to talk about that, we're really going to need to hit 3 main things. We're going to start with mechanism. That's critically important and underlies everything, so we're going to need to get a reasonably good primer on mechanism, talk about what neurovascular coupling is and how that has anything to do with what we measure with functional MRI. And then the next piece, of course, is measurement. How do we actually assess neurovascular coupling in vivo, in people? And we're going to look at some real data, both from aging and research on multiple sclerosis, and look at how functional MRI data relates to behavior. And we're going to end with a section on modeling. And we're going to be having an eye towards the future, talking about what is precision brain health, and are changes in a brain health index that's being developed here explained by changes in the HRF shape of a given individual. So let's start with a primer on mechanism. We're going to get the biochemistry out of the way first. So as some of you may or may not know, this is glycogen. Glycogen is one of the main ways that we store energy in the body. It stores glucose molecules in long chains. And as opposed to triglycerides, which are more long-term storage for sugar, glycogen is a more short-term store. And while this is found, particularly in muscles and in the liver, and even in your brain, the neurons in your brain, the ones that are the cells that are actually doing, what we would probably call computation and thinking, they do not store glycogen. So this sort of begs an important question of, neurons have to undergo metabolism like any other cell. How are they going to get the energy to do that? And the answer lies in what I sort of talked about in the outline slide, which is this process called neurovascular coupling. And so we'll start with the vascular piece of that. So this is a schematized version of a very small part of the circulatory system. On the left, you can see an arteriole. On the right, a venule and a network of capillaries that connects them. And you've got erythrocytes, red blood cells, that flow through those vessels. And oxygenated blood is flowing, being pumped by the heart, flowing away from out of the lungs, in our case, north, up, in a superior direction, toward and then around the brain. And it's carrying oxygen and glucose, but the oxygen it carries primarily diffuses at the site of these capillaries. And when oxygen diffuses, oxygen is carried by a molecule called hemoglobin within the red blood cells, within the erythrocytes. And hemoglobin has two primary species. One is oxyhemoglobin, which has oxygen bound to it, and the other is deoxyhemoglobin, which is not fully bound to oxygen. And this creates something that we leverage in functional MRI. It creates a natural and what we call an endogenous contrast. So if we look at schematics of the 3D shapes of these molecules, deoxyhemoglobin on the left and oxyhemoglobin on the right, we can see that there's a slight difference in the placement of iron. It sits slightly lower because there are unpaired electrons in the deoxyhemoglobin molecule. This is important for us using magnetic resonance imaging, MRI, to actually generate functional images of the brain. These unpaired electrons make deoxyhemoglobin paramagnetic, meaning that it will introduce slight perturbations and inhomogeneities into a magnetic field in which it sits, like, for instance, in the very large field of an MRI scanner. Oxyhemoglobin, on the other hand, doesn't have those unpaired electrons and is thus diamagnetic, meaning that it doesn't actually impact the signal, impact the field strength. And so we take advantage of this with MRI scanning to actually generate spatial maps of the signal that we're measuring in functional MRI, which is, we call it the bold signal, which is the blood oxygen level dependent. So what we measure with fMRI is fundamentally blood oxygen levels. And it's fundamentally located, the source of the signal is basically coming from the veins. But not just, we don't just use fMRI to get spatial maps. We also look at how the signal changes through time, and that's something called the hemodynamic response function. So this is a basic impulse response function that in response to brief stimulation, you will actually get what looks like a impulse response function that peaks about 4 to 6 seconds later and falls to an undershoot and eventually returns to baseline about 20 seconds later. And this is a time course evolution of the blood oxygen level in tissue. Again, primarily originating from veins, and I'm going to tell you why that is in just a minute. And so, importantly, blood is cytotoxic to neurons. So even though blood provides the oxygen and the glucose that neurons need, it doesn't interface directly with those neurons. So there's actually an entire network of cells that you probably all know better as the blood-brain barrier, which is predominantly made up of astrocytes. And so these cells are actually mediators, and they mediate the exchanges, and they mediate the signaling and relaying of needs in terms of metabolic needs to blood vessels that then return a vascular supply of blood containing the oxygen and glucose necessary for metabolism by brain cells. And so when we think about this in terms of a time scale, we can go all the way down here to the neuronal level. Conceptually, we can do that. With fMRI, we're not here yet. This is happening on the order of milliseconds. And metabolic changes happen about an order of magnitude slower than that. Neurovascular coupling happens an order of magnitude slower than that. And only when we get to the order of about seconds to tens of seconds do we actually start to see what we measure with functional MRI, which is the arterial, well, in this case, it's the venous, really. We can also measure the arterial component. That's something I'm going to talk to you about in a second. But this is, the venous part is really talking about how deoxyhemoglobin concentration is changing over time. And that's happening 3 or 4 orders of magnitude greater in time than actual neural activity. So we've got some distance. We've got a little bit of distance, a little bit of a gap that we have to bridge. So let's just do a quick recap on the mechanism, because I've thrown a whole lot at you. fMRI, functional MRI, we measure BOLD. BOLD is blood oxygen. Just remember that. Neurons do not store glycogen locally, so neurovascular coupling is the process that delivers requisite nutrients and metabolites as a supply in response to demand. And then the hemodynamic response function that I'm going to call the HRF, that it illustrates the process of neurovascular coupling over time and shows change in blood oxygen in the veins over time. Okay, moving into measurement. So the neurovascular coupling system can actually be measured in using a similar schematic, or we can envision it with a similar schematic to what we just saw, with the exception of we can't get to the neuronal level quite yet, and that's just a reality of technology. MRI does not have that kind of temporal resolution yet. So we already know that we can interrogate the venous component with BOLD. We can say, BOLD will show us what's happening at the level of the veins. Unfortunately, we can't use BOLD to inform the arteries, because remember that I said BOLD is a fundamentally venous phenomenon. It's happening in your veins. And that's because the veins possess a reasonable amount of deoxyhemoglobin. The arteries, the blood in the arteries is almost entirely oxygenated. If you've ever had a little, they're called a photoplethysmograph, a little pulse ox put on your finger that, or I think they have apps for your phone that do it too, that can read what your O2 sat is. That's basically trying to figure out what your oxygen level is, and it should be in the high 90s, if not 100. So we don't have, it's too, the contribution of any deoxyhemoglobin in the arteries is too small to image with BOLD. So we need something else. As luck would have it, people much smarter than I have developed a technique using MRI that's called arterial spin labeling. And this, let's break it down, because it's a bit there, so let's just break it down. Arterial spin labeling is labeling spins of blood in the arteries. So when I say spins, that's the behavior, any proton that is in a magnetic field, we conceptualize it as spinning. And so we can change the direction, the orientation of these protons in a magnetic field. That's how MRI works. Basically what we do is we change the orientation of protons with radio frequency pulses, and those actually, because they return at different rates to a low energy state, we can create tissue maps using MRI. And so the same idea here, we're actually trying to label the spins of blood in the neck, in the arteries. So that just involves inducing an orientation change in the neck. We wait for a certain amount of time to allow the blood that was in the neck at the time it was tagged to displace, and then we can actually acquire a signal in the brain. It's a little more than that, though, because we're not just going to get signal from blood, we're also going to get signal from everything else that's there, like the rest of the brain. So we need a way to just focus on the contribution from blood. And so the way we do that is called a subtraction technique. So we get one image where blood in the neck is labeled, and we get another image where blood in the neck is not labeled, but everything else is the same. And when you take a difference of the unlabeled image is called the control image, and then you have a labeled image. When you take a difference, what should be left behind is only the contribution of those labeled spins of that blood that was labeled in the neck. And so that's called a perfusion-weighted image, and you can actually put that through some further processing and get to something called cerebral blood flow. That's how much blood is flowing in the arteries. So we've got two now. two of the pieces we need for the neurovascular coupling system for actually measuring it. I'm going to now introduce the third piece. So, you know, BOLD is blood oxygen. CBF, that's just blood flow. That's just how much blood is flowing through the arteries. I'm going to introduce a new term, CMRO2. This is the cerebral metabolic rate of oxygen. In a shorter form, this is how much oxygen is being metabolized by brain tissue. So, how do we get this? I'm going to get to that in a second, but first what I want to do is I want to show everybody how these quantities change and how changes in one of these quantities or the other affects BOLD signal. Because I think that's really important for being able to have an appreciation for how we're interrogating this system. So when there's a stimulus presented in the scanner, you have a, remember back to that time scale plot that showed that metabolic changes happen on the order of hundredths of a second, happen much earlier than changes in blood flow. So prior to the change in blood flow, you get an increase in this CMRO2 quantity, an increase in oxygen metabolism. And you're actually having greater oxygen diffuse at the level of the capillaries. What that means is that more, what we have here is a venule, and each of these individual circles, quasi-circles, is a red blood cell, is an erythrocyte. The bright red ones are full of oxyhemoglobin, and the dark red ones are full of deoxyhemoglobin. So when you have increased oxygen diffusion in the capillaries, more deoxyhemoglobin is making it through. And you don't yet have a concomitant increase in cerebral blood flow. So what that means is there's more deoxyhemoglobin in the veins, and that causes BOLD signal to go down. So BOLD signal is going down as a result of more deoxyhemoglobin. So let's reset. Afterwards, you get that, the hemodynamic response function that shows that blood oxygenation. That's happening due to upstream vasodilation. So the process of neurovascular coupling, those mediator cells, astrocytes and others, are signaling to the vasculature upstream of where the metabolic demand is happening and the metabolic activity is going on. It's signaling those blood vessels to dilate. And so more blood makes it through, more oxygenated blood makes it through, displacing deoxygenated blood, and because more of the deoxyhemoglobin is being displaced, that causes BOLD signal to go up. So, like I said, I still haven't told you how to calculate CMRO2. So, to calculate CMRO2, we're going to use an advanced form of MRI. It's called dual echo calibrated fMRI. This is a mouthful, I understand that. So let's break it down. Dual echo. What do we mean when we say dual echo? Arterial spin labeling gives us, informs about arteries and blood flow. BOLD, functional MRI BOLD, that gives us information about blood oxygen in the veins. So if we measure both of them together in nearly simultaneous fashion, we can get both at the same time. We can get venous changes together with arterial changes. And so, again, people smarter than I have figured out how to basically fit in it's what's called a second echo, and they can actually get two images in each time that you have a labeling pulse and you wait a certain amount of time for blood to displace. And again, the same strategy applies here, where you're going to subtract the control from the label to take out all the background stuff and only leave the blood, the part that you're really interested in. But now you additionally have BOLD that you're measuring at about the same time. So that's dual echo. What's calibrated? So calibrated, all that really refers to is we're trying to calibrate the signal by manipulating one parameter, in this case it's blood flow, without appreciably manipulating the other parameter. And the other parameter is this CMRO2 that we wanted to calculate. That's the neurometabolic piece. So the way we typically modulate cerebral blood flow in an fMRI scanner is to show people stimuli, right? They see a picture of something, they see a flickering checkerboard, they hear something, and so that is going to change the cerebral blood flow due to neurovascular coupling. What we want to do now is change the cerebral blood flow without touching the neurometabolic side, without touching the CMRO2. And what that basically means is we need a vasomodulatory, vasoactive substance. There are a few candidates we can use for that, but the easiest one is carbon dioxide. I'm sure most of you know it's not carbon monoxide, it's not poisonous, it's just what your body produces naturally. And so what carbon dioxide has the effect of doing is it dilates blood vessels, particularly blood vessels that surround the brain. Those are particularly sensitive to carbon dioxide. And so if you expose a participant, if you give them basically a gas to breathe that is 5% CO2, you can switch out. Normal air is about a little less than 79% nitrogen and 21% oxygen, and then you've got a bunch of trace stuff. If you switch out 5% of the nitrogen that's inert and not really doing anything with 5% CO2, you can actually induce changes, increases in cerebral blood flow. So now we're using that to calibrate our signal. So that is going to allow us to get to CMRO2. There's math involved in that. Big surprise. I'm not really going to go into the math too much. Suffice it to say, it's algebra. We're trying to get everything that we know about the quantity on one side and the unknown on the other. And so by doing this calibration experiment with hypercapnia, we can actually get everything on one side, and the second unknown, the CMRO2, actually drops out of the equation entirely, because we're assuming that delta CMRO2 is 0, that term goes to 0, the whole term goes to 1, and it kind of falls out. Then combining a stimulation paradigm with a hypercapnia paradigm lets you get to CMRO2. I'm happy to talk with the intrepid and interested among you about the equations and everything, but I figure that most of you are probably like, okay, math, cool. All right, so we've got 3 out of the 4 now. This is good. 3 out of the 4. The last piece is this mediator piece. So if we think of neurovascular coupling as a 3-tier system, where tier 3 is the cerebral blood vessels, the arterioles and the arteries, and tier 1 is the neurons. And tier 3 is measured with CBF, and tier 1 is measured with CMRO2. Tier 2, the glia, and other cells that may be involved in neurovascular coupling, the ratio between these two quantities is going to inform us about what's going on. Because ultimately what this ratio really is we're saying that the ratio is change in blood flow per unit change in metabolism. So it's that index of the supply-demand relationship. And if these colors, the color scheme looks at all familiar to you, that's only because it is. So this, again, is our conceptual schematic model of neurons in tier 1, blood vessels in tier 3, and then the mediators, the middleman cells, astrocytes and others, that form the blood-brain barrier in tier 2. So now we know how to assess the neurovascular coupling system, how to measure it. So let's look at cases where we've actually done that. So my dissertation work that Dr. Rittman mentioned earlier actually looked at applying this dual echo calibrated fMRI technique in aging. And we had a research question that involved, basically the question involved are there age differential changes in task demand that have an effect on any of the, like the neurovascular coupling system at large, or any individual component of it? And so we did, we scanned older and younger people using hypercapnia, and then also using this visual task paradigm where they had a flickering checkerboard at 3 different frequencies. It was a yellow-blue checkerboard. We were trying to really maximize the contrast. And what we saw was, this is an exemplar participant. And what I want you to focus on in this screen here, this slide, is 3 areas. So in red, you have the focal area. In yellow, you have the proximal area. And in green, you have the distal area. And when I say focal, proximal, and distal, that is talking about the intensity of the signal that we measured there. So the signal was most intense in the focal region, and then that radiated out to the proximal and distal regions. And this is not terribly surprising. It was a visual task, and so this is your visual cortex right here in the back of the brain, the occipital lobe. And so we looked for main effects of age and frequency, but also the thing we were really interested in was interactions. And as it turns out, only a single quantity showed that interaction. And that was the neurovascular coupling ratio. So that's reflecting what's going on at the level of glia and other mediator cells. And so what you can see here in this plot, the 3 rows represent those 3 areas, focal, proximal, distal, from top to bottom. And then the left 2 columns show you younger and older adults, respectively. Those are plots of their blood flow on the x-axis and CMRO2 oxygen metabolism on the y-axis. So we're predicting CMRO2 from CBF and from age and from frequency. And what you can see is when we assessed this neurovascular coupling ratio, and we did that by taking the reciprocal of the parameter estimate that we got from this model, what we saw was that in the focal, so the highest signal area, what you might consider the most activated area, we saw this strong interaction wherein younger adults' neurovascular coupling ratio actually increased with increasing frequency in the task, but that dropped off pretty quickly, whereas older adults had pretty consistently flat neurovascular coupling ratios, regardless of how much tissue we were actually looking at. Okay, so moving on to modeling. So first thing I want to do is talk about MS. So this is more real data. And multiple sclerosis is it's a neurodegenerative disease, and it's often called a demyelinating disease because the body, for whatever reason, misrecognizes myelin, which is that substance that kind of coats the axons. The axons are kind of like the cables that connect your neurons to one another. If you think about it like that, they're not literally connected at the end. There's a synapse, but they're basically the projections. And the myelin sheath is this protective barrier. So it's like this is sort of like the copper, and this is the plastic on the outside of a wire. And so it it acts to insulate the signal. In MS, that's highly damaged and dilapidated. And the cells that are responsible for that are also the cells that are responsible for neurovascular coupling. So MS gives us this really interesting and unique window through which to view neurovascular coupling in the healthy brain as it compares to neurovascular coupling in a disease where it is very obviously compromised. And so we did just that by scanning MS patients and healthy controls. Now, I went into a lot of how we assess the neurovascular coupling system. The thing about that is, and it may come as little surprise to all of you, it's very involved to do that. There's a whole lot of extra steps that have to be taken. You need to do a hypercapnia experiment. You need to use a dual echo paradigm. You usually have to use a block design paradigm and get a lot more data because it's more subject to noise. And so for these various reasons, there's a lot of challenges to using that. And so we have another way to assess neurovascular coupling dysfunction. And that, if we go think back, the hemodynamic response function, right? That's assessing, that's describing quantitatively how neurovascular coupling is happening at the level of the veins through time. So we scanned MS patients and healthy controls that didn't differ significantly in age while they were performing two tasks. The first one is not terribly relevant here, but the second one is. So that's the digit symbol substitution task. It's an executive task. What we're really trying to do is we're trying to assess processing speed, and the way we do that is we present, it is based on a pretty gold standard measure of processing speed from the Wechsler Adult Intelligence Scale, but it's been adapted for use in the fMRI environment. And so participants are going to indicate by a button press, because they can't, fill out a form, a paper form in the scanner. They're going to indicate by a button press whether a key pair matches a corresponding pair a probe pair matches a corresponding pair in the key. And it probably helps to show you an example. So here we have on the top a 9-pair digit symbol key, and then a single test pair here below that. And the object is for the participant in the scanner to respond by button press whether this probe pair matches the corresponding pair in the key. I'm sure most of you by now have figured out that yes, indeed, it does. So they would press the right button. If it didn't, they would press the left button. So we did that while they were being scanned, and we modeled and generated hemodynamic response functions for everybody. And these are affectionately known as spaghetti plots. I will leave it as an exercise to the audience as to why we call them that. But what we basically saw was we looked in 3 areas. We looked in precentral cortex, that's motor cortex, right here. Occipital cortex, we heard of that already, that's visual in the back of the brain, and then prefrontal, that's towards the front. And that's the executive, reasoning, decision-making, working memory, a lot of things happen there. A lot of higher level functions happen. And so what we saw was that HRFs reached significantly lower peak amplitudes in prefrontal cortex, so in this plot on the right, in MS patients. And they took significantly longer to do so in both prefrontal cortex and in motor cortex for MS patients while they were performing this digit symbol substitution task. But an important question is, okay, that's interesting, but how does it relate to performance? We know that MS patients have impaired processing speed. That's one of their biggest complaints. How does it relate to performance? How does it relate to their actual measure of processing speed? And as it turns out, we tried 2 ways. We tried peak amplitude, and we did not find that peak amplitude of the HRF was able to predict performance. But time to peak, so peak latency, how long it took the function to actually reach that peak, was able to successfully predict it, but only in the MS patients. So in the MS patients where we had known neurovascular coupling dysfunction, because that disease acts as sort of a model condition for that, we were able to predict performance from a single measure of this hemodynamic response function, which was really encouraging. So the takeaways from this part of what I've been talking about, more canonical HRFs should reflect healthy neurovascular coupling. And a canonical HRF is one that behaves in an ideal way. So I actually took this part out because of time constraints, because I was going over. We did another study similar in older and younger adults, and we could not find a single HRF measure that was able to predict performance there. It's important, though, that we didn't have any measures of neurovascular coupling system integrity. And then in this sample of MS patients, even though it was a smaller sample, where neurovascular coupling dysfunction is known and it's basically a model of that, we were able to predict performance from a measure of this hemodynamic response function. So this gives us a lot of encouragement as to how we can use the hemodynamic response function in a way to sort of not just interrogate neurovascular coupling, but also what that means for more real-world and everyday considerations. Things like reduced processing speed, things like impaired working memory, those types of things. Behavioral consequences. Okay. Finally, I want to talk about, this is sort of with an eye towards the future. So this is precision brain health. So the goal of precision brain health is, for any given individual, we want to accurately predict longitudinal patterns of brain change that are specific to that individual, thus precision brain health. And one way that we at the center are trying to do that is by the development of this brain health index. So the brain health index is one that it's rather comprehensive, and it encompasses multiple factor scores that each represent different brain relevant domains. And there's three primary ones. Clarity, which is our domain for cognitive health. Connectedness, which is more social in nature, and then emotional balance and well-being. And the way these were derived was from a factor analysis taken from measures of individual sort of subdomains of each of the larger three super domains, I guess. And what we did was, this was work by my collaborator, Jeff Spence, and of course, Sandy, to actually examine and basically try to find the factor structure, try to find what these domains are. Go from, we've got a bunch of different individual assessments, individual tests that each tell us one thing, each tells one piece of the puzzle, and to construct a more complete index. So clarity, the one that I'm going to be talking about for the rest of the talk, deals with abstraction, big picture thinking, memory, sleep, and compassion. What is the utility of this brain health index in precision brain health? The answer is a little bit complicated, but the first thing we need to mention is that alone, the brain health index, at the end of the day, is, even though it's a measure of multiple brain-relevant domains, it's still purely behavioral, purely coming from assessments of behavior. And so what we really want to do is validate that that is not just measuring what you might consider, might be phrased as mental health, but really actually measuring brain health. And the way we do that, we're not at the point yet that we can actually say that this is validated, because that takes a lot of data. But we're on the right track, and we've taken a really, what I would describe as a really important and critical first step towards validation. And And so what we've been doing is we've been collecting neuroimaging measures on a subset of the people who are enrolled in the BrainHealth project, which you're going to hear about again at the end. And that is going to give us sort of this initial foray into attempting to try to make a good faith first attempt at, or first step towards validation. So once again, we can see the HRF here, the hemodynamic response function, right? This is describing changes in blood oxygenation over time. We've got three measures here. We've got amplitude, peak amplitude. We've got full width at half max, which is basically just how wide the peak is, measured at half the height. And the time to peak, that's the peak latency. But we actually added additional measures because we wanted to look at more than just these three. So we have a measure of, there's two additional measures. One is curvature. So curvature basically just assesses how much does the function curve, how much does it change direction. Mathematically, it gets scary because there's integrals, but it's not that bad. You basically take the second derivative of the hemodynamic response function, you square it, and then you take the integral over that, over the entire time course of the HRF. And what that gives you is a single measure that says how much this function curves. The second one is the integrated square difference. And so it's another integral. So we take the square of this deviation. So this deviation is our measured HRF minus the canonical HRF. And so the canonical HRF is this idealized HRF, which we just saw. It is, every parameter is set. There's only one free parameter to vary in a canonical HRF, and that's the amplitude. Everything else is completely set in stone. It's locked. And so what this does is takes the difference of those two, squares that difference, and then takes the integral over the entire HRF. And what that gives you is how similar is the measured HRF to the canonical. How close are they to a canonical shape? And both of these were normed. I just had that on there, but they were normalized. One additional insight that we had. We wanted to ask, what's the predictive power of HRF metrics, not only as they change through time, but what's the predictive power of baseline HRF metrics? What if something's happening at baseline? What if something at baseline is going to be informative and predictive? And so we had 10 total parameters, change in baseline for each of peak amplitude, time to peak, full width to half max, and curvature. We had the integrated square difference, which is the change in how canonical the function was, and then this last one, which is the baseline version of that. So we called that the baseline estimate of the non-canonicality of the HRF, which is a mouthful. So instead of saying that, we're going to call it benchmark. We kind of just added mark. Bench by itself sounded a little boring. And benchmark just it rolled off the tongue. So that's what we did. Okay. Previously, we tried to just predict behavior from a single HRF measure at a time. So what we did this time that was different was we trained a neural network model. So we had our feature vector, subsets of our feature vector that basically served as inputs to a model that had 15 nodes in the hidden layer and then a single node in the output layer. And the single node, basically, what it was trying to predict is, does a person's clarity score, or based on the features that we have fed it, can the model successfully predict whether a person, an individual's clarity score, because remember we're talking about precision brain health, that's the goal. Can it predict whether an individual's clarity score goes up by a certain amount? which we thought was a pretty interesting question. We used five-fold cross-validation. Basically, what you do is you split up your data set into 5 buckets, and then you train on 80% on 4 of those buckets and test on the 5th, and you do that for each combination. And so what that does is that's really useful for when we have smaller samples. That lets us do more with a limited amount of data. The accuracy of the model, 90%. The best model, the accuracy was 90%. So we successfully predicted, in the best model, it successfully predicted whether an individual would gain substantially in this clarity score between time 1 and time 2, 90% of the time. And that was from change in HRF peak amplitude, change in HRF full width to half max, and benchmark. Just those 3 parameters, those 3 features. Getting low on time. I have a little bit of a story to go through there, but I tell you what I'm going to do. I'm going to skip these for right now and come back to them if somebody asks me about them. And so what I'm going to do is I'm going to explain this. This is, if you haven't dealt with probability surfaces before, it probably looks daunting. It's really not. All this is anywhere you see very, very light yellow, that's saying the model is going to predict that a person is going to increase in their clarity score. Anywhere that you see a really dark red area, that is saying the model predicts that that person is not going to improve. And so we have change in full width to half max on the y-axis, change in amplitude on the x-axis, and then benchmark is each one of these individual panels. And so something really interesting happens here. For low benchmark, and low benchmark, what does that mean? That means that they're much more canonical. So they have a very high degree of canonicality. That means that that's going to result in having a low benchmark because the difference is small. For cases where they have very canonical HRFs, it's almost entirely a story of amplitude. So for people whose amplitude goes down, meaning that the function doesn't go as high, like it actually goes lower, they are predicted to improve. And the opposite is true. So people whose amplitudes actually go up, they're predicted not to improve. But that's only true when you have a starting place of high canonicality. For people who are less canonical, there's an interesting second path, second way that emerges for increases in clarity score. So what you see at the top for those low benchmark is that the right side of the plot is almost all dark red. But once you get to high benchmark, meaning low canonicality, it opens up this entire other part of the function, wherein if you have concomitant increases in amplitude and full width to half max, in other words, they change together, and they both increase, or in some cases both decrease, but particularly in this case that they both increase, that will also lead to a substantial increase in clarity score. And I just thought that was so interesting that based on whether you are starting out canonical or not canonical, the road that you take, or the road that your brain likely takes, that we're only seeing, after the fact, that is, you basically got a tale of 2 possibilities. And so with that, I'm going to go ahead and conclude. Bold fMRI. Remember, bold blood oxygen. The bold that we measure with fMRI, it's a composite reflection of an intricate, highly complex process that reflects at its core a supply-demand relationship, in that vascular supply is provided in response to neurometabolic demand. When we model HRFs without shape assumptions, or with very minimal shape assumptions, those are better able to capture variation in the health of the system. You can't do that when you use just the canonical HRF. but more complete characterization of each individual component of neurovascular coupling. It's possible, but it's much more challenging, and you have to use a more advanced form of fMRI to do it. Okay, I threw a lot at you, but at this point, I will take questions.
Okay. So we promised you a deep dive, and this is among the deepest of deep dives.
I hope
that
I
you'll
deliver.
see here at the Center for Brain Health, just to give you the idea of just how deep the dive can go. And so I wanted to actually start out with a question, sort of broadly for you, Monroe.
Absolutely.
So one fundamental mystery is sort of emerging in both MS research and aging research is that there seem to be some people who kind of over the course of the disease or over the course of aging sort of seem to kind of skate along across the top of the performance function. They don't change much. And others seem to really experience the adverse effects of either the aging process or the MS process. And so I was wondering how this work broadly addresses those issues of individual differences in changes over time or changes with a disease course like MS.
Yeah, that's a really great question. There's actually work that's been done from people I've worked closely with that has shown that if you break out MS patients, and this is specifically for MS work, if you break out MS patients into what you might call performance impaired, meaning that their performance is not nearly what it is in a corresponding healthy control, and then a performance preserved group where their performance is very close to a matched healthy control, that the neurovascular coupling system, the ratio is much lower. So the system is actually much more quantitatively impaired in the performance impaired group than in the performance preserved group. And that's the single measure that emerges as informative about the performance differences when you split them up that way.
Great, thanks.
Yeah,
So, I saw a question here. You had your hand up.
On a practical application, by doing these studies, does that mean that you will pick who should be trained more and for improvement and who won't benefit, so should fall out?
Yeah, that's a really good question. I think that is a goal of precision brain health, as far as the like in terms of being able to inform not just who should be trained, but like better tailoring cognitive training to fit individuals. That's definitely an application of that. And then what imaging lets you do is really, as we saw today, get under the hood of the changes that you're probably experiencing, or a given individual is probably experiencing. But what this lets you really do is have some explanatory power as to why that's happening. But that's a great question.
Okay, Sandy, do you have a question?
Yes. So my question is, we're hearing so much about inflammation in the brain as a kind of an earlier precursor of many diseases. And you think about inflammation affecting the neurovascular coupling, what would you anticipate this may help us to understand that, and particularly different drug trials or things that people can take to reduce neuroinflammation, which is so bad for the brain?
Yes. So interestingly, inflammation is one of those really challenging things to image because we haven't yet found a really good way to non-invasively image neuroinflammation. You can do it when you apply things like what's called GAD, which is short for gadolinium. So that's a contrast agent that you can give someone, and that will do a much better job showing inflammation. But it does create a bit of a challenge in sort of the more non-invasive, which all the approaches I talked about today are at their core non-invasive. And the most invasive thing is somebody has to breathe a gas that's 5% CO2, which is not in any way dangerous. And so I do believe my hypothesis would be that neuroinflammation would absolutely wreak havoc on the neurovascular coupling system, just because of how many processes and sort of subsystems are coming together, and they have to basically work in almost perfect harmony and perfect concert to achieve this kind of just-in-time delivery. I would say that it would surprise me if neurovascular coupling wasn't one of the things most affected in neuroinflammatory conditions.
Hey, another question back here first.
Yeah, great talk. I'm not a math guy. I'm an engineer, so sometimes it's more dangerous. In traditional feedback system, we take into consideration of the integral and the differential. So I'm wondering if you have looked at the rate of change of the curve, taking into differential as an extra matrix. I mean, it's very interesting that you look at the integral part.
Yeah, the main reason we wanted, and so the main reason we wanted to look at the reason we did the integral was just to better characterize or more summarily characterize like a single metric. And so we did actually, so one of the measures, this curvature measure, actually is the second derivative of the HRF. And then what we did is we integrated that second derivative. And so it does, well, it's been squared, of course. So we square it to remove sign effects. But so it's not exactly just taking the derivative, but I think it's close in spirit to what you're talking about. But what you're sort of proposing is taking the raw first derivative, right? We haven't done that, but that's definitely an interesting course to look at. We did the second derivative. It mostly had to do with the fact that we've modeled the function using splines, and we wanted to. It's an interesting thought. I think that's something we could try. Yeah, I appreciate the comment.
Okay, we're starting to run a little short on time. I want to get to, I'm just going to try to combine two online questions here into one. So you mentioned sleep earlier, and so there's been a lot of emphasis on sleep and the importance of sleep in neural processes. So what I want to, the question, and I'm going to try to synthesize this, is it possible that, I guess one part of it is, do you have any sense of how the role that sleep plays in the in these kinds of changes, especially the individual differences you've been looking at? And is it possible that that the specificity of the population that we're looking at, people who are willing, who come in for the BrainHealth project in particular, are among a healthier group that might be attenuating our ability to see individual differences and be able to look at measures like sleep?
So to your first question, it's definitely an interesting question. I will say that in the projects that I presented today, with the exception of the most recent BrainHealth project, which we are collecting sleep quality data on, the prior ones, the aging work that I did as part of my dissertation and the MS work, we don't really have measures of sleep quality, and so that's a really, I think, important question, and one that probably should make it into assessments and ways to sort of quantify so that you can explore for relationships to sleep quality. And there may be extant work already being done by people looking at the neuroscience of sleep, although I don't know that literature as well. To your second point, I think what you're getting at there is a bit of sampling bias, and it's possible. We're basically trying to do our best to get the most representative sample that we can. In any study, I think you have to keep in the back of your mind that there's a effective motivation, right? Not everybody in the world is going to want to participate in studies. So there is some degree of that, but there's not really a good, clear alternative of you have to, we mandate participation in studies. So it's, I think it's a good point and one that we need to keep in the back of our mind, but I don't think, I think in spite of that, we're doing a really good job of trying to.
I would agree to the extent that you've been able to show individual differences so far, so we're getting at least as representative enough to be able to see these kinds of differences.
Right.
Steve, couple minutes, got to move fast.
That's what they always say. So for those of us that play in the kiddie pool as opposed to the deep dive,
Okay.
is the better analogy here oil to a car engine or gas to a car engine in how important brain blood flow is to the operation of the brain? Because what's missing in this is the impact of the glia on the efficiency of that transfer, number one. And number two, is this a sign of efficiency or inefficiency, that blood draw demand?
Okay, that's a really great question. Jeremy, is there any way to put the slides back up? I was just going to show one thing that might better. Thank you so much. Okay, so just really quickly, participant 1 and participant 3 were both predicted to improve. No, I'm sorry, participant 1 and participant 2 were both predicted to improve. Successfully predicted, I might add. And what you notice for participant 1 is that the amplitude actually drops. And so that is a reasonable potential explanation, a reasonable hypothesis of why we're seeing what we're seeing, is that the system is behaving in a more efficient manner. There's a little bit of difficulty in assessing exactly why it's more efficient, but it's, I think, sort of hypothesis generating in that way, where we say this could be strongly indicative of a more efficient system, and then what you do is you more deeply plumb individual aspects of that to try and figure out exactly what components of the system are behaving more efficiently. I would say probably oil is the better analogy than gas, because what you're talking about is a system that is catalyzing. Like metabolism inherently is all about reactions. And you're talking about a system that is acting as a catalyst so that requisite reagents are available for those reactions to take place. So better analogy is oil in a car engine as opposed to gas.
Okay, so I think we got to end it there. Let's give Monroe a big round of applause for a really great talk. And lifelong brain health, this is our shared mission, and what we are doing here at the Center for Brain Health. So stay tuned. Our next speaker will be Jeff Cummings, September 22nd, talking about Alzheimer's disease, drug development, and what's new and what's coming.